Yes, so first of all, thank you very much for inviting me to talk today. Of course, I would have loved to come to Vienna again. Last time I visited, it was really a great time. So, and there's a lot of people that I would love to meet. But anyway, yes. So thank you. I'm very honored to have to talk to you today. So I changed a little bit my title. I turned the estimation into investigation because really that's what we're doing. We're trying to investigate what selection left on the genome of our favorite species. And so I worked in the animal genetics division at INRAE. And so although I develop methods and I also analyze different data sets, as Andreas was pointing out, in different species. And so I wanted today to talk to you about what we found in some of these data sets and how we investigated the selection signatures in these species. Okay. So the context, really, what I will talk about today is one of the possible ways of studying the genetic determinism of complex traits. So what we want to contribute to is to understand how does the genetic variation between individuals contributes to their differences in phenotypes, to these complex traits. And in particular, I would like to know which genetic variants are important for a particular trait. And so there are different ways of doing this, and one is to take an evolutionary perspective and to ask which variants contribute to traits that are related to fitness, so the ability of individuals to reproduce and to bear reproducing children. So when I'm talking about investigating selective effects, the idea is we want to identify patterns in genomic data that are consistent with selective selection. And we want to try to see if we can know which variants have been affected by selection in the past. Also, maybe which populations, at what time, and can we quantify the effect of selection? So I'm not saying we can answer all these questions rigorously now, but that's the goal. And I will explain how one of the ways we can try to do that. So when we're talking about identifying variants that affect complex traits, maybe the most obvious way to do so is to do QTL mapping or GWAS. So here the idea is that we want to find these regions on the DNA where we have variation between individuals at genetic level that produce a difference in the trait mean, and that we can detect when we sample individuals from a population. So when we do that, we really have this kind of modeling idea that the trait Y is linked to the genotype, to some causal functional effects. Maybe at the QTL, but there are also, of course, other QTLs, other variants in the genome that affect the trait, which we could call polygenic effect, and then some non-genetic factors. So that's one way of doing it. And I want to mention that when we fit this kind of models, we need to specify a model of action of the genotypes on the trait, right? So usually we'll assume there is some shift in the mean, but that's a linear model. Okay, so the other approach is to say, okay, now we'd like to know, maybe we can tackle this by looking at variants that affect thickness traits. So what does it mean? It means that, do you see my mouse, actually? Okay, hopefully you see my mouse.
Yes, we see it.
Okay, so if we have a trait that is actually linked to the fitness of individuals, then from one generation to the next, we'll have selection, and it says only some of these individuals in the population will reproduce, and they will tend to have biased genotypes because, say, this genotype is more fit because it has 2 copies of an adaptive allele, then the next generation, the distribution of the genotypes in the population will change. So that means that if we are looking at traits that affect fitness, then the phenotypes are going to affect the genotype of individuals in the population over time. So we're kind of reversing the previous model, where the response is on the genotype, and we want to know what are the effects of the trait on this genotype at a particular marker. And we also have other effects that affect our genotype. And these other effects can be other selective pressures, such as drift, mutation, or migration. Okay, so but this little sketch is to say that, well, selection leaves genomic signatures at UTLs, and we should be able to detect them. But if we want to do so, we also need to specify this kind of background effects on the trait, on the genotypes, just as we do in GWAS. Okay, so what are these selection signatures? So I guess you have, maybe some of you have already seen that kind of sketches, but nevertheless, so the kind of idea is that we have a population where we have different alleles that segregate here in a small genomic region. And then if one of these alleles, the red one here, is adaptive, then it's going to increase in frequency due to selection over time. So if you have a population that is selected, that adapts, then the red allele will increase in frequency. Maybe it will reach fixation, but maybe not. And so the genetic diversity patterns that happen around the mutation leave a signature, so what we call selection signatures. And the idea is how do we find those? So there are different approaches to do that. One is to look within the population, and we suppose we only have contemporary individuals of this population. So we only have the genotypes or the alleles in the population at a present day, and we want to find the selection signature. So we have actually somewhat little information to do that, but we can still identify some events. And the idea in these methods is to identify regions where you have reduced diversity around the selected mutation. So that's one possibility. So you have more extreme allele frequencies around selected mutations. And the other also signature that you can try to find is that you have longer haplotypes that are shared between individuals in the population. So you have these extended haplotypes. Okay, so that's one possibility. Another possibility is if you have multiple populations that have had different adaptive histories. So in this case, for example, here you have a population where you have selection and a population that is neutral. Then in this case, what you expect is to have increased allele frequencies differences between populations. And so you can test that. So I will be talking essentially about these approaches here today. And then a third possibility that I know many of you are familiar with is to look at the evolution of allele frequency through time. Because if you have selection, then you have a kind of a deterministic, if not deterministic, at least consistent evolution of allele frequency through time. And that can also be identified from data if you happen to have data over time. Okay. So today, the main questions I want to tackle is to see how do we detect loci affected by selection using this differentiation approach, and maybe what are the characteristics, what can we tell from the data we've analyzed. And I will be showing data from farm animals, livestock. These populations are actually interesting from a selective point of view. First, because, well, they are managed by men and they are selected by men for agronomical traits. And so the agronomical traits that we measure and that on these populations are actually fitness traits. individuals that are better for these traits tend to have more offspring due to human intervention. There is also usually a quite clear-cut population structure with very differentiated populations and not too much gene flow between them. Also because of man management, they tend to rear them in breeds that are genetically close. We have genomic tools that are available to study their genomes. And they also have an extensive selection history from the domestication from wild relatives to their recent breeding and improvement. So they are kind of, in some sense, models of adaptation. Okay, so to detect selection from population differentiation, we need samples from multiple populations. We need to reconstruct their demographic history. So that has to do with this R, this residual that I was showing before, where we really need, if we want to avoid drawing false conclusions, we really want to have a good model for their neutral history. And then if we can do that, then we can identify regions where this differentiation that we can kind of fit genome-wide does not match this demographic. Right? So genome regions where this like genome-wide history doesn't seem to fit. And that can be actually doesn't seem to fit in terms of they are outliers. Technically, we're going to be using goodness of fit of the neutral model. So is the neutral model good? If it's not, then maybe that's because there's selection. Or also, we can use models that explicitly include adaptive effects. And in this case, we will be testing an alternative. So is the alternative model of selection better than the null model of neutrality? Okay, so first I will present what is our model for the neutral evolution of neutral divergence between populations. So to do that, let's look at things through time for the beginning. So we consider the allelic locus, a SNP, in a population that evolves under pure drift, and it starts at some generation with an initial frequency of P0. And then because of drift, you have kind of this random process through time of the evolution of allele frequency. If we were to consider many loci that start from the same P0, same ancestral allele frequency, and we repeat this process many times, we will have some kind of distribution of possible allele frequencies for this loci through time. And actually, this distribution can be approximated by a Gaussian distribution. So this Gaussian distribution depends on two parameters. So the distribution of the allele frequency after the amount of time t depends on, of course, the initial allele frequency. And on average, they will stay at this frequency. Well, the average of the distribution is the initial allele frequency. And the variance here will depend on the amount of time that it increases with time. But it increases with time, but it also increases when you have smaller population size. So really, this variance parameter here is something that depends both on time and the effective population size. And it's 1 minus 1 minus 1 over 2n to the power of t, which is pretty well approximated when t is much smaller than n by t over 2n. And so provided this ft is not too large, we can actually model the allele frequency at time t by a Gaussian distribution with average P0 and variance ft P0 1 minus P0. This model was proposed, I think, in this form first by Nicholson et al. Okay, and does it work? Well, this is an example with simulated trajectories. In gray, you have the histograms with ft increasing, and the red is the Gaussian approximation. And you can see it's a pretty good fit. Actually, it's a pretty good fit as long as you don't keep the borders, so 0 or 1. If you have fixation probabilities that become important, then this model is not good anymore. Something to keep in mind. Okay, now we are looking at a single population. What happens if we have multiple populations? Well, if we consider an ancestral population here, and that there's a split at some point in time between different multiple populations, and they evolve in parallel, we get something like a star-like tree. And if we assume there is no mutation after the split for each population, and there is no migration between populations, then we have the previous model that is true for each of these populations. And if we were to make some even stronger assumption that all of these populations have the same effective population size, and we are all looking at the same time, same generation time, then we could say they all have the same Fi, and this is what's usually called the genome-wide FST. So people use the statistic FST to detect selection signatures sometimes, and you can see here that it's problematic in the sense that it's really making very strong assumptions. It's implicitly making very strong assumptions about the evolutionary history of this population, namely that they all have the same effective population size, and they're all splitted at some point in time, all splitted at the same time. So we know this is probably not true for most of the population we're looking at, and that's why we can actually come up with a model that's a better model that allows for more deviation to this kind of simple history. And the idea is that we want to allow the population to split not all at the same time, but sometimes you can have splits that are later, that happen later. So you have a proportion of the time where two nowadays populations were actually a single one. And so that's one of the trajectories that you would get, meaning that in these two populations, A or B or 1 and 2 here, you have an amount of time where they had the same allele frequency trajectory. And if you do this, well, what happens is that you get a multivariate model for P that incorporates a covariance matrix F. And this covariance matrix F, its values are the actual amount of time that was spent during the branch length of the tree here, right? So the unit of the time in unit of drift that was off diagonal that is shared between two populations. Actually, starting from this tree, you can relax a little bit this hypothesis by allowing for migration and covariance that are due to migration rather than just ancestral sharing due to being a single population. But the basic model is this one. And so F, this covariance matrix can be estimated from genome-wide data. And if we know F, we also can estimate the ancestral allele frequency by appropriately weighting the branches in our estimation of the ancestral allele frequency. So given F, at each SNP, we can estimate P0. So if we do that, then we have our neutral model. Right? We have a model under drift, more or less migration too, of evolution of the allele frequency. And so we can say, okay, at each SNP, we can test whether the observed allele frequency actually match this model. So really the model is included in this F matrix. And so we can compute the deviation, the observed deviation of our allele frequency to what is expected under this neutral model. Technically, this is a deviant statistic, and that needs to be a little bit transformed, but I won't get into detail here, to get what's called the FLK statistic that was developed in my lab by Maxime Bonon a while back now. And under the null, what's nice about this statistic is that under the null, that there is only drift in the evolution of the SNP. This statistic follows a parametric distribution, a chi-square, n-1, where A is the number of populations. So this is nice because we get p-values essentially. So we can actually apply a rigorous statistical significant testing which is very important. So later, what we did is that we extended this to account for linkage disequilibrium between markers. The idea here is that we use linkage disequilibrium models, so statistical models that describe the diversity of haplotypes in the population. And so that a given position here, we can say we have this, so we say there is a certain number of haplotypes, and we can then affect an individual, know which haplotype carries an individual. So we're transforming our single SNP test into multi-allelic test, essentially. And here you have a representation of the haplotype frequency in 4 different populations. And so, again, in the same spirit as the FLK test, we can say, does our distribution of haplotype frequencies in our population match the neutral expectation? And this is what was called the APFLK statistic, and that was developed by Marianne Fariello while she was doing a PhD study in the lab. And we showed that this statistic had good properties, that it had increased power in some situation over the FLK statistic. Okay. So this set of methods, then we applied it to different data sets. And I wanted to show you one of the studies that we've done in the sheep, because this is a study where we had quite a few populations, and we also used rather dense genotyping. So we could actually investigate in more details what was happening behind the selection signatures we can find. So let me talk to you a little bit about this data set. So it's 27 sheep populations that were sampled in France. So not all of them originate from France, but all the individuals were sampled in France. This is the population tree. On the basis here, it's an Iranian wild population that are the descendant of the original population that was domesticated. And so you can see that consistent with this history, this Iranian population is an outgroup, and the ancestral domesticated population should be here. Here you have a European mouflon population. So these are domestic sheep, ancestral domestic sheep that are found in Corsica and that have gone feral. So they are wild again, but really ancestrally they were the first representative of the domesticated sheep in the Neolithic, at the early Neolithic. And so you can see again, consistent with that they branch very deep in the tree here, and they are really not related to our modern breeds of sheep that are here. And in these modern breeds, you have really 2 groups that correspond to geographical differences. So you have, unfortunately, the south is on the top and the north is on the bottom, but really here you have the northern population and the southern population. I won't describe them in details now, but I want to point out 2 things here. You can see here a population with a very long branch length. This population is the Mérinos from Rambouillet, and Rambouillet is a town close to Paris, whereas there is a royal herd, and these sheep were offered by the King of Spain, I believe, to Louis XIV, or maybe Louis XV, anyway, a long time ago. And they were kept in close herd for since then. And that's why they have this very long branch length, because they have a very small effective population. And from this population, and this is another Mérinos population that has much higher effective population size. And you can see that there is this evidence taken from this trimix approach of an introgression from the Mérinos, possibly Rambouillet, into breeds from the north of France, I mean ancestors to the northern breeds, today's northern breeds. And this is possibly related to something that is historically documented, which is called the Mérinization of sheep breeds from France, where in order to improve the wool production of breeds, of French breeds, they were crossed to merino sheep that have very good wool quality. Okay, when we analyzed this data to look for selection signatures, we identify more than 100 outlying genomic regions in different places. So I won't go through all of them, of course. But just to say that there is decent power to detect selective events in this population. So one of the ones I want to talk about is an example of what we believe is, which would be called in a fancy manner, an adaptive introgression. So on one of the sheep chromosomes, we identify a very large signal for selection in both the northern and the southern breeds. And here you have a representation of the local haplotype diversity in these different breeds. Here you have mostly populations in red that are from the south, and they all share the same haplotype. So there is a very strong selection signature here. But you can see you have these two populations, Ile-de-France and Berrichon, that also share the same haplotype, although they come from the north. And so what we believe here is that this already adapted haplotype was introgressed during this event into the northern population, and that was later selected again in this population. So this introgression of an adaptive allele. In this region, the problem is this region is very large because the signal is so huge. And there are two candidate genes. One is ABCG2, which is involved in the protein yield of milk. But maybe more likely is this L-Corel locus that I will talk about later, that is very strongly associated with stature in different species. And another interesting thing here is that you can see there is another haplotype that's been under selection in another breed, the Manège-Tétrus, which is a breed from the Pyrenees. But it's a different one because it has a different color, so it's a different set of alleles. And that's something that we actually could pinpoint in many of the selection signatures that we find, is that there is strong evidence for allelic heterogeneity. That is, on the same locus, so we found many different adaptive elements. So how we did that? What we did is that we considered the signal that we can find of selection in one region, and we decomposed it and tried to find how many independent events or alleles are necessary to explain the correlations between the allele frequencies in the tree. And so we used a method that was actually initially developed to look for the number of EQTLs in EQTL studies, but we can actually apply it in this case. And what we find is that although we have many selection signatures that seems to correspond to a single allele being increasing in frequency in the population, we have quite strong signals for allelic heterogeneity in many of the signatures. And in particular here, you can see you have one of these regions where you have a lot, like maybe 6 or 7 possible alleles segregated. So what happens here, it can be different genes in the same region, or it can be different alleles of the same gene. This region here is a region that confers baldness, so the absence of horns, and it's possible that it's actually been selected, recurrently selected, and multiple alleles confer the same phenotype. But we can also, because we don't have really sequencing data, it's also possible that if you have very old mutations, many haplotypes carry the same allele, so we are not completely sure. But in one of these situations, what we did is we actually resequenced the gene that we knew were under selection in this region to try to find out if we could see this allelic heterogeneity, and this was the case. So this is a gene called MC1R that you may know because it's involved also in the hair color in humans, and it's involved in coat color in sheep. And we have 3 populations that have black face, so they have black coat color. And these 3 populations are different haplotypes. And when we resequenced the gene, for one of them, we could identify actually that it was carrying a known mutation in the coding region of the gene that is known to confer black coat color. But the 2 other populations apparently carry different variants. And these are not coding variants, they are regulatory variants. So knowing how they work is a bit more complicated. Okay, so this all I would say about the sheet data set, but the key thing is that we can find selection signatures. There seems to be extensive allelic heterogeneity, and that we confirmed it on one of the examples. We can actually improve the inference when we use sequencing data that we don't always have, but we did do that when with my colleague Simon Boitard, and when we studied this 1,000 bull genome data, so the full resequencing of 4 cattle breeds. So there are only 4 populations, a bit of the limitation in this case, and they are quite distantly related. So in this case, you see the kind of this star-like shaped tree is a good approximation of the evolution. When we conducted the analysis using as if we only had SNP chip genotyping or sequence data. Here I'm showing the PP plots. And we can show that we have much more power with sequence data than we would have with only SNP chip genotyping. We found a lot of different selection signatures with different approaches, and we were able to show that using sequence data, we were able to actually pinpoint the actual causal mutations when we knew them. So again, this gene MC1R also responsible for coat color in cattle. There are 2 mutations that confer either red or black coat color in the gene, and they were actually the top signal in the signature. The other example is this gene called PLAG1, and PLAG1 has been shown by genome-wide association study, or by QTL study really, to be responsible for polymorphisms around this gene have been shown to be responsible for the stature phenotype in cattle. And the author could pinpoint a few variants that could be the causal ones. There's a handful, 5 or 6, and they were actually the top candidates in our selection signature work. So that shows that using sequence data, these selection signatures can either pinpoint causal variants. And what we found is that if we took the top candidates, even the top regions in all our selection signatures, all of them, but the one on MC1R, were outside coding regions. So that indicates that these data actually are consistent with the fact that selection acts most on regulatory variants than on coding variants. But once we've done that, we don't really know. So first, they are regulatory, so we need functional data to meet what kind of regulators. Are they promoters, enhancers? I don't know, long coding RNAs, and for that there is currently a lot of work being done to functionally annotate those genomes. So we will have to revisit our study after that. And also, what are the traits that have been under selection? Because this approach don't tell us what traits. So what we can do is we can compare what we find with QTL studies. So that's one possibility. And so that's what we did in another round of sequencing of cattle, and where they did a so in this paper, our colleagues did a meta-analysis of 160, and they found 163 QTLs for stature in cattle, and we could show that 50% of them were actually in selection signatures, and that was a large enrichment compared to if they were just random, it was just overlap by chance. And here I'm showing you 2 of these selection signatures on 2 genes that I already talked about. So the LCORL gene, I talked to you about in sheep, but it's also a QTL for stature in cattle, and it's a very strong selection signature in cattle, and PLAG1 that I just talked about. And here you have these 15 populations that were studied, and you can see that almost all of them have selection signatures in at least one of these 2 genes. So a selection for stature. The only one that doesn't is the Jersey breed, which is a very small breed of cattle that is raised on the island of Jersey, and that is has a high yield because you can put a lot of animals on the same field. So it's very small. All of these other breeds are pretty large animals, and most likely they are large because they've been selected on one of these genes. I think one of them has both of them, and it's the Charolais, which is a very, very heavy cattle. Okay. So, but again, this is just, so we are using GWAS to inform us. And we would like to be able to maybe incorporate the trait in our analysis. And that's been proposed a while back by Graham Coop. And the idea is that you collect additional data on your population, so their phenotype, they can be direct measures, you can do government garden experiments, or in our case, you can actually use the records that are available for them. And they can also be things that are linked to fitness to adaptations of bioclimatic variables, like temperatures or parasites. And in this case, what you do is that you include your trait in your model. So you're testing an alternative model for the evolution, where you say, okay, the value for my trait actually has an effect on P. So there is a link between the trait value and the genotype in this sense. And we can actually incorporate that and derive new statistics that test this alternative against them. And so what we wanted to do is to do a simulation study to see to what extent this allows to be to detect selection better. So what we did is we simulated a population that evolved in a fitness landscape. And this fitness, so it's a map, it's kind of the Landy model, it's a typical model where you have the fitness, you have an optimum for fitness, and then you can be outside of this optimum with a certain variance, and this variance kind of defines the intensity of selection. And we imagine a population that is outside the optimum and that evolves toward this optimum by phenotypic selection. And we simulated actually selection in population trees with different fitness landscapes. So one that I call smooth is that the actual value, optimal value for the trait is highly, two populations that are closely related genetically also share optimum values for the trait. So you have to imagine populations that adapt progressively in a kind of a smooth way to new environments, say going north. Right? So going north adapted to colder climates. And another scenario where you have no relationships between the optimal value for the trait and the genetic relationship between populations. So you can have two closely related populations that have to adapt to very different optimums. So that can happen, for example, if you have adaptation to altitude. You have low geographic distance, but very high difference in the optimal value for the trait because the environment changes very rapidly with distance. Okay. So, and the question we wanted to look at in this simulation is what's the power to detect these QTLs under this varying scenario? So we have our trait model. So our trait is determined by a QTL, the one we're going to investigate, and some other genetic effects. So it's a polygenic adaptation, but we will be focusing on one of the QTLs. Okay, so we're simulating a polygenic trait with a particular feature that we will focus on. When we do selection on Y in this model, what happens, it's sketched here, is that over time, so if your optimum doesn't change and you have something that's very neutral looking, if you select for an optimum of, say, 3, well, anyway, if the unit of variance is 1, what happens is that on average your allele frequency increases, but your population adapts by increasing the proportion of, adapt through the QTLs, but also through the polygenic component. And so you don't fix the QTL usually in this situation. And actually you have a large variance in the response, and maybe sometimes even your QTL becomes maladaptive, because if you're already adaptive for the trait, then maybe increasing this trait too much becomes negative for the fitness in this model. So, and the other thing that we wanted to look at is the population structure. Is to which extent the fact that you have highly structure versus no structure affects your power to find those QTLs that adapt, these adaptive QTLs. And so what we did is that we started from a very structured tree, a binary tree that was a random binary tree. And then we merged internal nodes sequentially until we had none. So we have kind of a measure of population structures, which is the number of internal nodes in the tree. Here, if we have 4 populations, we have 3 internal nodes at maximum, and then 2, then 1, and then no internal nodes, and we have this kind of this star-like shaped tree. And so we want to see what are the effects of the fitness landscape, the population structure to detect QTLs, and also parameters that are linked to the QTL itself. So this is an example of the simulations, just to show you a bit how it works. So here it's with 8 populations. Here, so this is 2 well-balanced trees, the same structure, but the fitness value here is rubbed. You can see these 2 populations, they are closely related, but they have very different optimum values, -5 and 5, right? So what happens, and this is the smooth one, where when you are closely related, you have close optimum values. What happens in the rugged landscape is that, for example, if we focus on these 2 here, you can see that when they split and they have to adapt to very different optimum, then the allele frequency, the mean phenotype adapts, and the allele frequency also diverges very quickly. Whereas in the smooth landscape, so we have here trajectories that kind of look like the tree, but they can be quite divergent. Whereas when we have a smooth landscape, when you look at these trajectories, you can really see the population tree, the neutral structure popping out. And this is true at the mean phenotype level, and this is true at the allele frequency level. So eventually, we still, in both situations, we have a clear relationship between the mean phenotype and the QTL frequency. But in the smooth landscape on the right, we also have a clear relationship between the distance between the genetic distance between population and the mean phenotype. So we have kind of a confounding between structure and adaptation, whereas it's not true in the rugged phenotype case. Rugged landscape case, sorry. So we simulated under these scenarios with varying QTL allele frequencies and effects. We took 20 populations to have rather large samples. The covariate we took were the average final phenotype in each population, and we tested with differentiation tests, FLK, AppFLK, or the one that incorporates the phenotype that are called FLK-adapt and AppFLK-adapt. And we varied the population structure as I explained. So what happens in this case? So first, what happens if you change the genetic architecture? Well, what we can expect that when you increase the proportion of the variance in the trait that is due to your QTL, well, you increase your power. So when your QTL is larger, so either it's larger or it's more frequent, because this proportion of variance explained depends on the QTL effect and the allele frequency. You increase your power. You can see that when you include the trait, when you test specifically for the adaptation for the trait, you actually have much more power than when you testing only do the allele frequency diverge. So including this information is improving power. Okay, but you can see that when the QTL has a small effect, then it becomes very difficult to detect. Okay, so polygenic traits, hard to find this QTL. But what's maybe more important is that this effect actually, and this difference in power actually depends also on the population structure, how much relatedness there is between population and the fitness landscape. So let me show you through these pictures. So here on the x-axis, you have the number of internal nodes. When you go over, so when 0 is on the middle, so that's the star-like shaped tree, there is no structure. And when you go on the right, you have more internal nodes, so more population structure under the rugged landscape. And when you go on the left, you have more population structure, but under the smooth landscape. So, and this is in a situation where we have power with adaptation test. So what happens if you're on a rugged landscape, population structure doesn't matter very much, right? You can have population structure, given that it's completely unrelated to the value of your trait. It doesn't affect power that much. Adaptation traits, testing adaptation is a better idea if you can, but the difference in power is not that large, and it doesn't vary much with structure. However, when you're in a smooth landscape, right, when you have very confounding between adaptation and neutral drift, then the differentiation tests, they lose power very fast. Whereas adaptation tests, they tend to retain power a little bit, although you see it decreases too. Okay. So what that means is that really our ability to detect selection signatures is something that depends on complex factors such as the population structure, so the demographic history, but also the ancestral adaptation, the fitness landscape that the population has had to adapt to. Yeah. So we also applied that in sheep, but I see that I'm a bit above the. So just to tell you quickly how we do that on real data. So for sheep, on the previous data set I showed, we have access to the population average of the traits that are used in the breeding of the sheep, in their genetic improvement. So we could use this data and also some morphology traits that you can find in databases, and you can come up with a score that kind of tells you whether a breed is a tall and large or whether it's a small breed. Right? And so using this, we can take many traits together that kind of describe the morphology of the animal. In this population, we have kind of something that's smooth. There are these southern populations that tend to be smaller in stature, whereas the northern populations tend to be very big, and especially some of these breeds are used in crossbreeding. They are very heavy breeds, they are very, very muscled, they have large muscles, but you still have some counterexamples, such as this one called the Charmoise, which is a small breed from the north, or the Lacone, which is tend to be a taller breed, but that comes from the south. And when we did that, we can find selection signatures that we can relate to stature. So one is the myostatin gene, very well-known gene involved in the regulation of muscle growth, and two other regions where we know there are QTLs in cattle, but also in humans that are related to stature. And one of them we don't know. Okay. So just to go to conclusions and a bit of discussion. So this population differentiation approach, they can identify selection signatures in livestock population, and looking at them, we can reveal the selective history of adaptive alleles. When we use sequence data, we can see, and this is not the only example where we have this evidence, that most of these signatures fall in regulatory regions of the genome, so we need better annotation of animal genomes. We have strong evidence for allelic heterogeneity at adaptive loci, and also we have shared loci across species. So I talked to you about LCOL, MC1R, but there are other genes that seem to pop up very frequently in different species, including not farm animal species. So we would like maybe to say there are selective hotspots, but we have to be careful when we say that, because maybe they are just the loci that we have good power to detect. And that's kind of the point of the end of my talk, was to say that when we interpret the selection signatures, we have to be very careful about the fact that our power to detect those are very affected by the genetic architecture. So we have very little power to detect selection on polygenic traits. So that doesn't mean that they are not selected on. One of these traits is, for example, milk production, milk yield in dairy cattle. We have very little signature that we can relate to milk yield, but we know it's under strong selection. So it's probably very polygenic, and we just don't have the power to detect the loci that are involved today. But it's also affected by the population structure and the fitness landscape of this population. And this is things, so population structure we can recover from the data. Fitness landscape is more complicated. So we have to be careful. So this means that we need to be careful in the interpretation of the results, and in particular, that kind of limits the generalization of findings today. So how do we go forward and try to avoid these things? I mean, do better? So we could work with polygenic adaptation models. I've seen in your seminar you had a good talk about these models. But you also, I think, have heard that these models have problems when we have the estimation of SNP effects that are biased due to structure. And in livestock, we have very structured populations, so it might be difficult to get very reliable individual SNP effects that can be used in polygenic adaptation models, although we will try. We believe that exploiting functional annotation information can be useful and can help in pinpointing the true causal variants and maybe the true gene. And we want to work on models that are genome-wide, where we consider all the loci jointly, not only for estimating the neutral model, but also for estimating the selection coefficient. And the last thing is something we've been started working on is genetic pathways. I didn't have time to talk about that today, But using genetic data available in time can also be very useful and provide better power to detect those small effect criteria. I would also like to mention the work by Buffalo and Cook that showed how looking at covariance between loci can be very useful in detecting polygenic adaptation. And this is a good, also in our context, it's very relevant because we have experimental evolution of selection lines, a little bit like you could do with evolve and resequencing experiments. We have also now a large cohort of individuals that are genotyped over time due to the genomic selection programs, so selection programs that use genomic information. So we are accumulating genetic data over time, and now it's been more than 10 years for some of these species and some of these populations, so we can start looking at time series there. And also the possibility to work with unchained DNA and there to study adaptation over longer time frames, such as domestication. Okay, with that, I would like to mention people that have been working with on this subject. So on methods, Maxime Bonhomme, Marie Nesfariello, Simon Boitard, and my colleagues also Claude Cheval and Magali San Cristobal. And also for the data analysis, Christina Roques, which was a PhD student in the lab. My colleagues from SHIP, working on SHIP, Carole Moreno, Isabelle Fallière, Jérôme Raoul, and David Torturo. And also Gwenola and Florence Clisson-Petit, and also partners from the 1,000 Bull Genomes, in particular Didier Boichard and Ben Ace, that are members of this project. Thank you for your attention, and I will take any questions.